Hey, my name is Tobias and I'm a postdoc from the Department of Neuroscience here in Sheffield. And today I'm here on behalf of the organising committee for the departmental research meeting to interview Kate, who won the NC3Rs prize. So if you'd like to introduce yourself.
Hi, I'm Kate Whittaker. I'm a research technician in the Department of Neuroscience, and I work in the Fly Lab with Ryan West.
Great. Okay, and could you start just by telling us a little bit about what you presented at this year's meeting?
Yeah, so I presented a poster about my project, which is it's exploring motor neuron disease and frontotemporal dementia by looking at visual dysfunction in flies with a form of those diseases, and measuring that dysfunction using electrophysiology.
Fantastic. Okay, and what were the key findings of the work you presented?
Well, it's early days in terms of findings, but what the project is about is looking at a particular genetic form of those two diseases. So motor neuron disease and frontotemporal dementia are on a spectrum. So they're linked genetically and in terms of how they present. And so there's a particular gene called C9orf72, and a mutation in that gene is the commonest genetic cause of both those diseases. And essentially what happens with this mutation is in the DNA, you get a long repeat expansion, it's called, and then that actually then makes toxic proteins. And so those are associated with disease when they form in neurons. So we think that an early sign of these diseases is visual dysfunction. And so I'm using flies that have been given this defect, these toxic proteins, a model that our lab has developed. And it's the only animal model actually that has got that defect in the same way that humans have got it, to the same length of protein. So it's a really good model. And I'm looking at, I'm exploring the effect on vision. So what I do is I get a fly, I put it in front of a special TV screen and I show it contrast stimuli. So like it might be black and white grids or grey and so on. And then I measure with electrodes the response of the neurons in the visual system to those patterns.
Lovely. Okay, that's great. And also I mentioned earlier in this interview that you won the NC3Rs prize. Could you maybe tell us a little bit about what that is?
Yeah, so the 3 Rs in NC3Rs stands for replace, reduce and refine. And it's talking about how we use animals in research. And so although it would be nice not to use animals at all in research, they are used, and it's really, really important part of being able to understand these horrible diseases and to find treatments for them. But the program, the NC3Rs program, is about ways that we can make that better. So reducing the number of animals we use, replacing the animals that we use with lower order ones, and doing experiments in a way that cause the minimum suffering to animals. And so the models that we've got, the flies, they actually tell us a lot about these diseases because the model is so similar to what happens in people. But it's better using flies than the alternatives, which are things like mice and rats and so on. So so we're trying to get that valuable information, but with a minimal impact on animals.
That's great. And what are the implications for your work moving forward?
Well, so what we're trying to do is understand better how these toxic proteins affect people who have motor neuron disease and frontotemporal dementia. And then what we can hope to do is, one thing is that we can use this system to test drugs that might make the effects of the toxic proteins better in patients that have got the diseases. And another thing that we can try to do is to take and so if we know how the toxic proteins affect the visual system, because surprisingly flies and humans have got quite a similar visual system, we can potentially spot the signs of motor neuron disease and frontotemporal dementia earlier. So we're looking to develop biomarkers, and that can be by scanning. So non-invasive, we can use this electrophysiology on patients.
Okay, fantastic. And or just final question then. So what is next for you in your research career? So what are the next stages moving forward?
Well, last year I did a master's in neuroscience at Sheffield, which I absolutely loved. And so this project for me is a great way of getting more experience in neuroscience research. I'd love to continue doing that. And so I'm hoping to get more experience in that. And so working in Citran in the Institute of Neuroscience in Sheffield is fantastic, and I'm hoping to do more of that.
That's great. Okay, all right, that's everything from me. So thanks very much.
Thank you. Cheers.